Hi, my name is Dr. Mousumi Majumder. I am a professor in Department of Biology in the Faculty of Science at Brandon University, and I'm a Canada Research Chair in Genotoxicology. So right now, you are standing one of the latest laboratory we have here at Brandon University, which is named as Breast Cancer Cell and Molecular Research Laboratory, and it is funded by Canada Foundation for Innovation, matched by Research Manitoba, and the vendors with their in-kind contribution. Gene and toxicology. So as we all know, gene are those genetic code present in every single cell in our body. You and me, all of us have the same thing. Here we are investigating what are the impact of environmental toxicity to change the genetic code, particularly small RNA molecule called microRNA. And we are interested in investigating this phenomena in case of breast cancer. First of all, we have the promise in that proposal that we are going to come up with the biomarker, which means that those are the marker can be found in your biofluid. For us is the blood plasma, and test that if those biomarker can actually distinguish cancer versus normal. If that is accomplished, then can we go beyond that and actually test that these biomarker which we just got, what are their roles in the genotoxicology? You are one of those who knows how to move forward with our knowledge and experience and investigate and finding to put forward Canada at the forerunner in the field. Every day I feel like that I have too much to do and I have to live up to the expectation, the honor I receive. And I'm glad to tell that this laboratory and this infrastructure is actually playing the pivotal role here to move forward with the research questions I have, to ask that question with that excellent support of machine and instrument, and increasing the capacity right now. With this Canada Research Chair, I have this CFI laboratory. Now I can train the next generation of Canadian scientists. It began with the Canada Research Chair recognition. I immediately got the notice that I also got funding to build this laboratory from CFI. And then I got the Research Manitoba matching funding. The provincial support comes with it and the vendors in kind. And along with that, we got recognized by Breast Cancer Canada. I'm lucky to be one of the 4 in Canada got funded. Earlier last year, we have a breakthrough in laboratory. We found out that these microRNA are doing something fantastic. The cancer for which you don't have much of a cure, incurable breast cancer or triple negative breast cancer, we have been working with a drug called doxorubicin. It's literally called cellular poison. So it kills growing metastatic tumor cell. Interestingly, we observed that these microRNA high tumor cells, when we fed them with this drug, it seems like that all dead, which is excellent. We see that after 4 months under the microscope, we can see that some cells are beautifully growing. They are drug resistant. And then if you identify that the mechanism, how they develop the resistance, so you can revert that back and make them resensitize to the drug. This is very, very exciting. And right now we have at least 4 different projects focusing on that drug resistance along with the biomarker, which is always our first priority. We now have developed and identified those small molecule called microRNA, and we found that they are very sensitive in detecting cancer in the blood. So we published that data right now, and that came out from this laboratory. So we showed that these microRNA can distinguish benign or tumor. So it can detect very early stage 1 when tumor is really small, treatable, manageable. So go by surgery, chemo, and radiation or triple combination. The very next step would be for us, we are going to develop a RNA-based blood test, which is going to be like a kit. They are going to be a test marker added to the list when patient goes for their physical checkup for blood test. Our goal is to bring those markers we are identifying in tumor model here to reach out to the clinic and make those markers listed as one of the markers to be detected as a screening tool. So next goal is to reach from this bench actually to the clinic. And we call it popularly that from bench to bedside medicine for breast cancer screening. That's the goal.